This is a Professor, CBMD PhD. And today's Wednesday, August 5th, 2026. It is 5:30 and 48 seconds, a.m. Eastern Standard Time. And today's research, Ruptosis. So let's hop to it here. Stanford scientists discover immune cells that explode, like microscopic BOMBs. Date August 5th, 2026. Source Stanford University. Stanford researchers discovered immune cells in flatworms that explode within minutes, killing nearby bacteria and foreign cells before vanishing completely. Their unusually fast and localized attack could offer clues for developing targeted treatments against infections and cancer. The discovery was made in planarian flatworms, small aquatic animals known for their extraordinary ability to regenerate, even when cut into pieces. These worms can rebuild missing tissues and, in some cases, produce complete new organisms, studying how their immune systems have survived and adapted over hundreds of millions of years, can offer useful clues for modern medicine. The researchers and scientific investigators named the newly discovered cells Ruptosis because of the dramatic way they respond to a specific hormone. They never expected that a cell could just explode like a BOMB and kill the cells surrounding it. The researchers first noticed the unusual cells while investigating a long-standing question in flatworm biology. Scientists wanted to know whether flatworms can distinguish their own tissues from those belonging to another individual. To test this, researchers slice worms lengthwise and fuse each one with part of a different worm. Flatworms are highly effective at rebuilding their own bodies, but these fused Frankenstein worms rejected tissue from unrelated individuals. The reaction resembled the way a human immune system may reject a transplanted organ. The cellular response, however, was very different from anything normally seen in humans. It is this huge inflammatory response, like there's a fire, an alarm goes off. Previous research into flatworm regeneration has shown that the hormone activin plays an important role in their survival. Elevated activin levels can weaken a worm's ability to regenerate, while low levels interfere with its ability to reproduce with other worms. As the fused worms begin rejecting foreign tissue, the researchers detected a rise in activin followed by chronic inflammation. The animals did not die immediately, but they perished within several days. They also found that rejecting activin into healthy, non-fused flatworms produced a similar inflammatory response. To examine what was happening inside individual cells, researchers used live cell microscopy and flow cytometry, a technique that uses lasers to analyze and separate cells. They marked the cells within different fluorescent dyes and then isolated those that reacted to activin. A small group of cells suddenly burst open, released substances that killed nearby cells, and disappeared within 5 minutes. The researchers named this explosive process Ruptosis. The speed and completeness of ruptoblast self-destruction distinguish ruptosis from other known forms of cell death. Some mammalian cells and bacteria may also do an explosive sort of cell death, but the time scale is really long. They are exploding, but it is more like pores that slowly leak things out over the course of several hours. Ruptosis happens within seconds to minutes. This rapid destruction appears to turn each ruptoblast into a highly localized weapon. Instead of slowly releasing harmful material, the cell uploads its contents almost instantly. Researchers tested ruptoblasts against E. coli bacteria, human kidney cells, and mouse blood cells. The ruptoblasts destroyed all 3 types of targets. The damage remained limited to cells located close to the explosion. It did not spread through a chain reaction or leave behind persistent toxicity. According to researchers, this ability to deliver a powerful but tightly contained attack could have implications for future treatments targeting bacterial infections or tumors. Ruptoblasts also differ from familiar immune cells such as T cells and neutrophils. Those cells are hematopoietic cells, meaning they are blood cells produced in bone marrow. Ruptoblasts are granular cells. The researchers believe ruptoblasts intensify their normal secretion systems so they can release toxic substances suddenly and violently after encountering activin. A rapid surge of calcium from the endoplasmic reticulum inside the cell helps drive ruptosis. Researchers and scientific investigators searched for similar cells in other species. They found them only in basal bilateria, such as flatworms. The restricted distribution suggests that ruptoblasts originated early in animal evolution. Researchers propose that vertebrates may have lost this defense because they cannot easily repair the surrounding damage caused by ruptosis. Flatworms, by contrast, contain abundant stem cells and can replace damaged tissues with remarkable efficiency. It demonstrates there is lots of different immune mechanisms out there. There is all these animals that live in an environment where there is lots of bacteria, lots of viruses, and we know so little about their immune mechanisms. The findings show how much scientists may learn by examining animals that are rarely used as traditional research models. Although flatworms may appear simple, their unusual biology can reveal immune strategies that are absent from humans and other vertebrates. The researchers are investigating a wider range of organisms that may inspire new approaches to some of medicine's most challenging problems. And this videography was created for educational purposes only. My code name is Will. I'm Professor Christopher Brunet, leader of research scientist, ufologist, psychic, computer technician, computer programmer, philanthropist, writer, mathematician, artist, photographer, film producer, chef, animal, and nature lover. My passion is world peace. Have a great day. This is a Professor, CBMD,